the Egyptians. So
That's enough, I
okay,
think.
okay. Officially now, we will start. Yeah.
Yeah.
So I'm the Hyung from the Inje University, and it's my great honor. And then I'm very happy to have my talk in this the webinar series. Because the Professor Prince is a very good friend of me, and then we can call each other as a brother. So a long time we cannot see each other, but something like that family reunion is today. And the topic of today, my talk is the cerebellum is a mobile cardiac excitation contraction regulator. And I'm working in the cardiovascular and metabolic disease center in the University, Busan. And actually, the professor Prince stayed in our centers more than 4 years. And then he had many publications and good research results. So today I will give some the contents. Is there some introduction and energy metabolisms and then cerebellum and ion channels and cerebellum study will be talking in these sessions. So as you know, the cardiac heart failure is the number one cause of the hospitalization, also the number one cause of death worldwide. In the worldwide, more than 26 millions of people are suffered by heart failure, but there's a more numbers are estimated the prevalence of due to the undiagnosed cases. And if you starting your the heart failure cardiovascular disease, then more than 50% of patients are re-hospitalized for the heart failure within 6 months of the discharges. So actually, that is one of five people are have some risk of the developing heart failure. So that is a really severe and really some life-threatening disease in the world. When we see the heart failure progress, there we can say that two types of different the heart failure. The first one is the ventricular remodeling after acute infarctions. So if you have some coronary occlusions or any kinds of the physical damage in the heart, you can start the initial infarct, some part. After that, the infarct is expanded. So the expansion of the infarct is from the hour to some days. They are increase their infarcted size, and they are going to the global removal in stage 2 months. So there is the left ventricle, general left ventricle is important for pumping the blood out of the heart. So they're getting the hypertrophy and dilated cardiomyopathy. And some of that other part of the heart failure is made by there are some kinds of the metabolic stress or some kinds of the unknown cause. So ventricular remodeling in the diastolic and heart failure is that they are starting the hypertrophy under there some hypertensions or there's some diabetic cardiomyopathies, some kinds of the metabolic stress. So the hypertrophied heart is starting from diastolic heart failure. Then they are go to dilated heart, so systolic heart failure finally. But in the both of that part, the calcium signaling, finally they lost their contractility in the heart. That is heart failure as a final stage. And in their heart contraction is very sophisticated, regulated by the calcium excitations and contraction couplings. So we can call it the EC coupling dysfunctions. The excitation is electrical, some signaling starting the contractions, and then contraction is the physical movement of heart itself. So when the heart is to give the some electric signals, calciums come into the heart by the calcium channels that can stimulate the calcium release from the SR. So this is calcium-induced calcium release. We can call it the CICR. And then there is a huge amount of the calcium released from the SR to the cytosolic part. And then they can start to the contractions, the series of the contraction mechanisms. So after that contraction, the calciums are reuptake into the SR by the circle, or it can be removed out of the cell by the NCX or calcium pump. So that is totally well the orchestrated the mechanisms of cardiac contraction and relaxation. But if that is some breaking point of this calcium EC couplings, they can cause the loss of the contractility and then become the heart going to the failure state. But when we check some kinds of the studies in the preclinical or clinical studies, so that alkaline calcium channel is very important, but the density and the regulation is altered in the failing human ventricle myocyte. And it's recovered after support with the mechanical assist device. But in their studies, even that the alkaline calcium channel protein is going down, the density is going down, the function is going down, but the alkaline calcium gene, mRNA level was not changed. So actually, that is the after transcriptional some modulations or the dysfunctional modulation is existed, not because of the transcription, but after transcriptional regulations. So what is the mechanism of the reduction of alkaline calcium channel? The patient with the heart failure is still the main part is unknown. So we move to the cerebellum. Cerebellum is a substrate recognition component of the DCX. And this is DDP1 code for Xbox E3 protein ligase complex. So this the E3 ligase complex is generally the role of that is marking on the proteins with the ubiquitins. So the marked protein with the ubiquitinated proteins are degraded by the proteasome ubiquitinated mechanisms. So if that is, then the CRBN has the role as the recognize their target. So the CRBN grabbing some of the target proteins, and there are some subunits or complex are labeling the protein to be degraded by proteasomal degradation process. So CRBN can find the target proteins, so that is why important. And until now, there is the known target is some kinds of ion channels, or very importantly, the AMPK gamma subunit unit was known as the CRBN target. When you look into the CRBN's some structures, either 2D structures, there is the 441 amino acid with the drug binding domain and lump substrate the binding domains. And this is the complex of the CRBN with the DDB. And there's some the pocket for the binding with the drugs and also the binding for the target proteins. So that is some the illustration after the graphing and the ubiquitinated by degradations. The first discovery of CRBN was the year 2004 from the US. There's some psychiatric doctors, family doctors. He met a family has some mild mental retardations family in the European families, and he found a mutation of the ATP-dependent lung protease gene. So they call it as a cerebellum because that was found in the cerebral cortex. So actually, they find these genes as some the neuronal or brain functional regulators. And our collaborator, Charles Bach, is actually the pioneer of the other CRBN studies. He identified this protein is working as the binding with large conductance calcium activated potassium channel in the left brain. And this channel is very important for learning and memory. And that is how can explain why that is something linked with mental retardation in the model. So how they find that there are BKCA channels and cerebellum binding is he is an electrophysiologist and he want to find some regulator of the BKCA channels, and then they do the EST2 hybrid screening. So by using these kinds of the techniques, he screening so many proteins and genes, and he finally found one genes is he called it as the BP1. BKCA channel binding protein 1, but is renamed as cerebellum. So when they started the suppression of the BK channel surface expression is by the cerebellum regulations. So finally, their first finding was cerebellum regulating the BKC channels in the brains, and they modulate their channel expression in the membrane. In year 2010, there is more very important finding was discovered by Hiroshi Honda from the Tokyo Medical University. And he found out the cerebellum as the thalidomide-induced teratogenicity target proteins. So what is this kinds of the thalidomide? Some the set, and that is kinds of the very sad event strategy in the drug development. So thalidomide was developed for as a immunomodulatory drugs in 1957. Germany invented for the morning sickness of the pregnant women. But after they take these pills, their children were born without their legs or their arms. So the severe teratogenicity was found in the children who their mothers take this pill. And worldwide, 12,000 infant was the Pocomelalia, and 50% of them is death. That is the recorded as the worst drug accidents in the world. But until the year 2010, there's the cause of these kinds of the accident was uncovered. But they are finding the cerebellum binding the thalidomide on immunomodulatory drugs, and that is why the children has their severe teratogenicity. So that is the first mode of action of the cerebellum. Actually, cerebellum should degrade some substrate, which is very important for the development. But with the drugs, their proper regulation of the degradation on time was inhibited, and that can make the kinds of the malfunctional development of the babies. In year 2013, there is one more very important finding was done by the person, but he found out the AMPK activity was regulated by the cerebellum. So what he did the studies, he found that the CRBN knockout actually increased the AMPK alpha phosphorylation, so that can enhance the AMPK activity. As you know, AMPK is very important for the metabolic regulators, and this can be good for some mimicking the exercise effect. So in their study, they found out AMPK gamma subunit is actually a regulator of the AMPK alpha phosphorylation. So AMPK gamma subunit is degraded by the cerebellum. So without the cerebellum, the AMPK gamma subunit is still there, and then they enhanced AMPK alpha phosphorylation. So increase of CRBN, decrease the AMPK activities. So when they making the AMPK, the knockout animal models and then cell models, they found that the AMPK alpha phosphorylation is significantly increased, and also their response to the metformin, the AMPK alpha, the phosphorylation activator was much better. And then when they making the CRB knockout mouse, they are the anti-obesity property against the high-fat diet. When they do the challenging with high-fat diet, they are relatively lean body weight compared to the wild type. And when they're watching into the liver, CRBN deficiency prevented the fatty liver in the high-fat diet mouse liver. So there is less the oil or fat depositions in their liver, even though they take the high-fat diet. And then there is some more the important finding was done in the year 2014. It's a Nature and Science. And they found that CRBN is actually another laws with the thalidomide binding, they can actually inhibit the cancer cell proliferations, or even they can kill their cancer cell, certain types of cancer cell. So actually, they are some of the blood cancers are targeting. So that is the anti-cancer effect is the another mode of action. So with the AMID, there is some substrate replacement. So the IKZF1 or 3 is Icarus and Iris was important proteins for the cancer, especially the blood type cancers, the survival and proliferations. But if that is there's without the drugs, there has a very low affinity to these proteins. But with the drugs, they are affinity is getting stronger and they easily breaking down the essential proteins for the cancer proliferation and survival. So with these drugs, it can working as the anti-cancer drugs. So until now, there's so many kinds of some their targets were discovered, like as BKCNA channel and chloride channel, ORI1 calcium channels, AMPK gamma subunit. In the cancer part, there's a glutamine synthesis, also known as the target of the cerebellum. And this, the panels, you can see that is the neo substrate. That is only, or they enhance the binding affinity with the cerebellum with in the presence of the IMID drugs. So that was these genes are the targeting for the limb development that can cause the teratogenicity. But the under this, the genes or proteins are important for the cancer survival. So the targeting these proteins and degradations are actually showing the anti-cancer effect. So anti-multiple myeloma or the anti-melodysplasia was the role of the cerebellum with the drugs. And also there is some kinds of the non-protein target was the suggested like some DNAs can be the target of the cerebellum. And there's also unknown substrate, but they have some function like a skin barrier function and cellular senescence. So let's move to the heart. When you're looking into the heart, because that is important for the some channel activity regulations, and also that is important for the energy metabolism, we are looking into the human patient. So when you're checking their NCBI GEO database, we found it that CRBN level in the heart is highly increased with the related cardiomyopathy patients. And that was confirmed with the real patients with a human heart failure. So that is highly increased with the low ejection fraction patient. That was the started with our collaboratory patient in Japan. So we can make the hypothesis cerebellum maybe can activate the advanced metabolisms and also regulate the ion channels like as calcium channels. So electrical stimulation and energy metabolism can be regulated by the cerebellum in the heart. So we used the cerebellum knockout mouse and then we compare their heart functions and their structures. So you can see the heart, skeletal muscle, all of that cerebellum was gone. And we can find out the slightly hypertrophied phenotype of the CRBN knockout mouse, their cross-sectional area and total area. And body weight is a little bit lean body weight than the wild type. So part 2 body weight is a little bit increased with the cerebellum knockout mouse. But actually, there is the AMP or BMP, that is a pathological, the marker of cardiac hypertrophy is not much changed. So we can see this is something like the physiological hypertrophy in this model. And when we checking the cardiac functions with echocardiography, we found there is a ejection fraction and fraction shortening was increased in these motors. And then when we do the ex vivo cardiac functions with the Langendorf system and pressure, the volume, the regulation was much higher in the cerebral knockout mouse. They can say that without the regulation of the brain or some neuronal regulation, that heart has a much bigger contractility. And that is the DPDT, and the stroke work was also enhanced in the knockout mouse. And then we come into the cardiac myocyte itself, because even in the heart, there's several types of the cells are consisted. So that is some the nodal or some the ventricle and atrial cells has their roles. So we really want to checking if that is a cardiomyocyte itself has the influenced or affected by the cerebral knockout. So in the single cardiac cells, we can find out their contractility was highly increased in the cerebral knockout mouse. And also, when we're looking into the metabolism of these animals, we found out the cerebral knockout mouse show higher energy expenditure, so the consumes more energy than their wild type animals. So something like that, they are to the mimicking the exercise. And we're looking into the mitochondria, and this is the heart, the mitochondria, and you can see more dense and much more numbers of mitochondria in the cerebral knockout mouse. The oxygen consumption rate was enhanced with the knockout mouse. And also the ATP productions, but there is the RH generation was lower in this model. So CRB knockout may be very helpful in the mitochondrial function and the quality control. Because the increase of their heart functions, we can think about that is energy metabolic the aspect and also calcium regulations. So we want to check if that is calcium signaling can be changed in these models. And this is the calcium signaling is measured by the computer microscope. So in the single cardiomyocyte, you can see the much brighter this wave. That means the much higher the calcium transient in the cerebral knockout mice. So there's a calcium amplitude is much higher, and this is the action potential durations. So actually, these kinds of plateau is prolonged by the much higher calcium entries. And we're looking into the alkyl calcium channel, some functional studies. Maybe if you are not so familiar with these kinds of the result, but you can see these kinds of peak base to the peak difference. So that is the cerebral knockout mouse. It has a much bigger calcium current than the wild type. So is that transcriptional regulations or actually binding the protein and degradation? Post-transcriptional regulation, we're checking their gene level and the protein levels. So you can see the CAV1.2, the coding genes in CAC and A1C was not changed, but only the protein levels changed. So let's say that is not the transcriptional regulation, but this the post-transcriptional regulations. So we want to know if that is a really directed target of CRBN is the CAV1.2 with the staining and the co-localization studies. And these two signaling is well overlapped. That means the cerebellum and CAV1.2 is located in the same location in the cells. So there is a high probability of the correlations or interaction between two proteins. And also there's some simulation studies if that CRBN can bind with the shape 1.2 with the computational modeling. So there is the higher possibility to the binding of two proteins. Then we check if that is really binding of that kinds of two proteins, so shape 1.2 and CRBNs we do the core IP studies. So you can see with the CIB 1.2, with the CRBN, they can be the well bind and then precipitated. Then we are cutting some part to know which the part of the CIB 1.2 can bind to the CRBN. So there is that the N terminal and C terminals and L1, L2, the part is we are separately do the IP studies, and we found that the N terminal of CIB 1.2 can be binding with the CRBN. And which part of the cerebellum is we found that is the loan, the CT, NT, and CT part. We found that the N terminal of the cerebellum loan binding domain was the binding site with the CIB 1.2. The binding is that can be also induced. So is that really the binding can making the degradation or ubiquitinations? And we check it, the ubiquitination of the CIB 1.2 by the cerebrons, and then we found that is really the ubiquitinated by in the presence of the cerebrons. And CIB 1.2, the N terminal is the ubiquitinated and then degraded in this model. And we also checking the effect of the thalidomide, which is the binding site is shared with the CIB 1.2. And if we treated of the thalidomide, that kinds of binding and degradation was inhibited. Then we do more study to the mechanisms because that is the whole body knockout mouse. So we want to do some more studies to clarify our data. So we express the CIB 1.2 channels with the hex cell, then we overexpress the cerebellum level. So cerebellum CIB 1.2, the current was decreased when we overexpress the cerebellum. So this one also the support our data. CRBN can degrade the CIB 1.2. And fortunately, we have the collaborator. They have some specific cerebellum degrader. We can call it the cerebellum prota, TD165 drugs. So they can use the VHL ligands. So it can specifically degrade the cerebellums. So in the HEC 293 cells, when you treatment with the thalidomide, so we overexpressed cerebellum in the HEC cell, and then we treated these drugs, and they are very effectively degrade CRBN levels. And because of this one is if that is really linking to the CIB 1.2, the biotilation, that means that the membrane expression. So we check that one, and then the TD165 actually decrease the cerebellum level and enhance the CIB 1.2 biotilations. So membrane expression was increased this one. And we also checking in the cardiomyocyte, the cell models. AC16 is a mouse cardiomyocyte cell lines, and also the CIB 1.2 biotilation was increased with TD165 cerebellum degrader. And we want to check if that is a really functional or some clinical relevance. So we do the doxorubicin-induced heart failure models, and then checking the contractility is preserved in the CRBN knockout mouse. So we found that the ejection fraction was much better than with the cerebral knockout mouse before and after doxorubicin treatment. And also we do another model with the MI myocardial infarction models by surgery. We got the same result with the different types of the heart failure models. Actually there, the contractility was better in the cerebral knockout mouse. And we also confirm if there is a really causative effect. So we overexpress over cerebellum if that is the kind of enhancement was removed by the overexpression of cereblon. So increase of the cardiac contractility was decreased by CRBN overexpressions. And this one is also we checking their membrane, their ion channel studies, and we found it actually cereblon overexpression decreased these kinds of anti-calcium channel activities. And because the reviewers are very tricky and they want more data with the cerebral cardiac specific gamma, so we make it. And we got the same data with the cerebral cardiac specific knockout mouse. So in this mouse, it's only the heart CRBN was knocked out, and then the cardiac function was enhanced. It's like as a whole body cerebral knockout mouse. So finally, we find out the cerebellum level is highly increased with heart failure with the reduced ejection fractions. And this increase of cerebellum degraded more calcium channels. They reduce the calcium channel density and their functions in the heart failure patient. So in vivo models, the cerebral knockout mouse has a much bigger cardiac contractility with the increase of the beta-calcium channel activities. So thank you very much for your listening. And if you have any questions or comments, it's welcome. This is the members who helped us.
First of all, I would like to thank Professor Kim Hyun-kyu for his inspiring lecture. And if anybody would like to ask a question, okay, Mustafa Tamami.